Great. Welcome to Seminars in Oncology. It is my great pleasure to introduce Dr. Judith Agudo, who will be speaking today. So Judith performed her graduate work in Barcelona, working on studying diabetes and how to get stem cells to survive in type 1 diabetes. And this really drove her curiosity because she realized that the thing that kept, plaguing the ability of these stem cells to engraft was really the immune system. And so after her graduate work, she realized she wanted to learn more about immunology and came to the United States for a postdoc with Miriam Murad and Brian Brown, where she developed a beautifully elegant mouse model looking at T cells that were specific for the model antigen GFP. And if you guys are thinking about this, you realize the universe of different mouse models that already have GFP incorporated in various cells, various rare cell types. And so she had some beautiful papers looking at the ability of stem cells, not just in the pancreas, but in many organs, to either be killed or not be killed by CD8 T cells. And so this naturally led her to be interested in CD8 T cell killing of stem cells of all sorts, including various cell types in cancer. So we were so lucky to recruit her to the Dana-Farber, where she's a member of the Cancer Immunology and Virology Department and has really been investigating this question of how does tumor heterogeneity and these sort of more dormant or stem cell-like populations in cancer actually resist or not resist T cell mediated killing. And so you're going to see a really elegant story today that's the culmination of her first few years as junior faculty. And I have to say, amazing as this work is, my favorite thing about Judith is the wellspring of kindness that she has for the people in her lab, for her colleagues, for everyone around her. And it is rare that I get to introduce someone who is scientifically so brilliant and such a good person and a good friend. And anyway, Judith, I will stop talking to allow you to have more time for your seminar, but I'm very excited to have you here today.
Um, Thank you so much. You shouldn't say those things about me because I can get emotional and I don't want to start my presentation crying. Anyway, thank you so much. This was a really, really nice introduction. So as Stephanie said, I started my scientific journey, not as an immunologist, but more of a, I would say, person into regenerative medicine, realizing very painfully and very early that the immune system was tremendously important in, right? Like if we try to regrow, regenerate or transplant something. And when I had to come to the United States for my postdoctoral training, it was very clear to me I needed to learn immunology. And back then I had a very naive question, which drove, right, like my research since then, which is this idea of phenotypic heterogeneity and how different cell types can either get killed or not get killed by T cells. And if I were to ask any one of you in the audience, if you were like close here, if we were in person, right? Like let's say we have a T cell that recognizes cell X, and the T cell is fully functional, activated, and it finds cell X. What's going to happen? And I'm sure that 99.9% of people would say, well, cell X is going to get killed. Well, are we sure? Because we have never studied every single cell type to know that that's actually true. We know that's true because we have studied autoimmunity, we've studied infectious diseases, but there are cells that we don't know. And I'm going to give you one example. That's the example that drove me here. So when I was studying type 1 diabetes, I knew beta cells were easily killed. And this happens in mice, in dogs, in people. Next to the beta cells, you have alpha cells that are very closely related, very similar, and they never get killed, ever. There's no such a thing as an autoimmune disease against alpha cells. So are alpha cells actually susceptible to T cell killing? And this naive thought brought me to ask, beyond alpha cells, what do we know about this heterogeneity? And even more so, what about stem cells? There was this idea in the regenerative field that, in the regenerative medicine field, that stem cells cannot get killed. But then when you dig in the literature, nothing has really been proven. So can we investigate that heterogeneity to learn how cells can escape, if they can, from T cell killing? And this has very deep implications, not only for regenerative medicine, which is obvious, but beyond that, for example, for cancer. Now we know, right, with cancer immunology, with cancer immunotherapy, that cancer cells don't get killed. And what cancer cells do is they hijack pathways that are already there, right? PD-L1 didn't show up to protect cancer cells. PD-L1 was there to stop immune responses, and cancer cells take advantage. So what can we learn from tissue heterogeneity? And the problem is, if I wanted to study, for example, are alpha cells susceptible to killing? There's no tool for me to study that. There wasn't. If I wanted to do this, what the immunologists usually do to study T cell specific responses against a cell type is they use what it's called a T cell receptor transgenic mouse. So T cells that all carry the TCR that recognizes some antigen that is specific for whatever cell type that you are interested. So for example, if I want to study killing of alpha cells that express glucagon, one thing I could do is develop a transgenic mouse whose TCR recognizes glucagon, for example. But then if I want to study killing against 20 different cell types that no one has ever studied, whether they get killed or not, I would need to make 20 different TCR transgenic mice, which was obviously not feasible, and for many reasons. So, right, like we were scratching our heads because we really wanted to answer this question. And then we realized that the answer was actually much easier. Because if you go to, for example, the Jackson Labs website, you find there are hundreds of mice that express GFP in almost any imaginable cell type, right? Like anywhere that you could think of, you could be interested of, it's there. This is actually an image. This is not from Jackson Labs. This is from the GenSat project that has, I don't know, 1,000 mice. I don't even know. So if we have this plethora of mice that express GFP very specifically in distinct cell types, then we thought, why won't we? I mean, we can just make a mouse whose T cells recognize GFP. And now GFP becomes a model antigen, and a model antigen that is present in almost any cell type that we would be interested in studying. And this is basically what we did. We vaccinated very quickly because this is published. We vaccinated mice against GFP. We got these anti-GFP specific CD8 T cells, and we cloned. So we took these T cells and we basically cloned a mouse out of these T cells, but by somatic cell nuclear transfer. And we call these mice just eGFP death-inducing T cell mice. For short, the Jedi mice. And once we made the mouse, of course, I mean, you can imagine, right? Like we took many GFP reporter mice that were available and started to try, right, to see what happens. The first thing was to try, well, do these T cells work? So let's get a cell, a target cell that we know has to get killed. And in my case, the one I knew very well were beta cells. So if you take mice that express GFP, right, under the control of the insulin promoter, so these are the beta cells, if you adoptively transfer Jedi T cells, what happens is all these beta cells get killed and the mice develop hyperglycemia. So this was very nice because this allowed us to show that these T cells could be used to develop models of autoimmunity. This was for type 1 diabetes, but you could do this for MS, for many other diseases. One great advantage that we didn't value until this moment is that GFP can be seen without the need of staining. So for example, this islet was not stained. This is straight, right? These are live cells. Another advantage is that when you have an antigen that you can see, this allows you to see even one cell that is alive and has escaped. So for example, this one beta cell here has GFP, is surrounded by CD8 T cells and still alive. And the fact that I don't have to stain to see the cell means that I don't have to fix, I don't have to kill the tissue. Now, potentially, I could get back this cell, FACS sort, profile, and see, right, how is this cell not getting killed? So this was another great advantage of this GFP platform. Something else we could do, you can study trafficking of T cells in various organs. So for example, here we could find that CD8 T cells very effectively are able to enter into the brain. So the brain is not an immune privileged site, like it has been many times claimed. T cells enter, go in and out. And in this case, what we did is to show that T cells can kill microglia. These are the microglia in the cells expressing GFP. And after JEDI, they get killed. And the third thing that we reported here in this work was that now with this GFP transgenic, this plethora of transgenic mice that express GFP in combination with the JEDI T cells, we had a platform in which we could deplete cell types and study the function of a cell type. So let's say someone has some reporter mouse that expresses GFP in cell X, and they want to know what the function of cell X is. You could inject JEDI T cells, cell X gets depleted, and then you go and see what the phenotype is. So it's kind of like nature's way to generate cell knockout mice. And in this case, the example was we have mice that express GFP in Tregs, so FOXP3 GFP. If we inject JEDI, Tregs get completely wiped out, and this phenocopies exactly what you see in the FOXP3 diphtheria toxin receptor mice. So it's another way to study. Of course, in this case, we know what the function of Tregs is. We could very nicely see inflammation and autoimmunity in these mice, but if we didn't know what FOXP3 cells do, we would have found out by using this depletion platform. Once the technology was published, we went into what was, right, like the interesting business. So I was sort of on the, I was searching for an immune evasive cell. I wanted to show that it's not true that a cell is doomed to die if there's a T cell that is activated, that the cell, when they encounter a T cell, there's always, right, like there's communication that it's not doomed to die. So I decided to go into stem cells, also because I had my background in regenerative medicine and I was interested, right? And it's a very relevant question, because if there's autoimmunity in an organ, if the stem cells in that organ succumb during an autoimmune attack, if you were able to stop that autoimmune attack, that organ can no longer regrow because the stem cells die. Conversely, if there's an autoimmune attack and the stem cells somehow have evolved means to escape from immune surveillance, then when you manage to stop the immune attack, that organ has a chance to regrow. So it is a major question, but it was a completely unanswered question, at least with regards to adult or somatic stem cells, the stem cells that we have in our bodies, which are the ones that can get attacked by the immune system during autoimmune diseases. So how do we study this? Well, if there's anything that stem cell biologists like, is to put fluorescent reporters in the stem cells. That's how they get to see them. That's how they get to study them. So what we did here was we took these mice, LGR5 GFP, that were developed by Hans Clevers in the Netherlands. And these mice express GFP in various stem cell types, and only in stem cells. So in the intestine, epithelial stem cells in the ovary, and hair follicle stem cells in the skin. And then we just injected activated anti-GFP JEDI T cells. And interrogated, what's the outcome? So the first stem cell that we studied was the intestinal stem cells, because we felt that They had to be important, right? These stem cells are here. These are your 5 stem cells. These actually were the first ones that Hans Clevers described with these mice. And these cells, what they do is they are constantly proliferating to replenish the epithelium in the intestine. So that sounds like a pretty important job. So if any of them had to be immune-evasive, we thought these would be the ones. And when we adoptively transferred GLI T cells, what we found is actually no. They get killed very effectively, very efficiently, super quickly. Okay, next one. Epithelial stem cells in the ovary. The ovaries are a very special place, and they sounded like they should be immune-evasive, right? Because they generate oocytes. What happens when you inject GLI T cells? What happens is actually the same. They don't escape, they get killed. So this sort of dogma or belief in the stem cell field that stem cells are universally immune-evasive actually is not true. Many stem cells die when T cells are, right, like targeted against them. Next cell type was these hair follicle stem cells in the skin. And this time when we injected GLI T cells, to our surprise, they didn't die. And this was the last one I was expecting to survive. But they didn't die. And a very long story made it super short. T cells go there. It's not a problem of trafficking. T cells are activated. It's not Treg dependent. So what's going on here? And we studied multiple stem cell types. They were all epithelial. They all were dependent on Wnt signaling because they all had LGR5. What was the difference between these stem cell types? And we realized that the major difference was that the hair follicle stem cells are extremely quiescent. They do not proliferate for weeks and weeks, while conversely, the intestinal stem cells and the ones in the ovary, those are super highly proliferative stem cells. So we thought maybe the cell cycle actually is a phenotypic feature that drives immune evasion. If that's true, if we get hair follicle stem cells that proliferate, then we could answer the question, and maybe when they proliferate is when they become susceptible. And if there's something great about the hair follicle is that they actually, right, like hair follicles go through cycles. They are in a resting phase in which the stem cells are extremely quiescent, but then from time to time, they enter into a growth phase and then the stem cells start proliferating. So what we did is we caught, right, like we got mice in the phase where they are proliferating and then injected GLI T cells. What did it happen? Well, what happened was that now we got a lot of killing, lots of stem cells died when they were in this proliferation phase. And we found out that the mechanism that allowed them to survive when they were quiescent was downregulation of MHC class 1, but not only MHC class 1, it was downregulation of every single gene that matters in the antigen presentation pathway. And it's not that the genes were zero, they were all a little bit expressed, but every single one very, very low brought the pathway to such a, like low level that it was insufficient for T cells to recognize. And this regulation was completely linked to the cell cycle. So when cells went into cell cycle, they upregulated class 1. When they were into quiescence, they downregulated class 1. And this was the last work I did as a postdoc. So when I finished this, I had these two big questions that brought me to start my lab. One of them was, well, if stem cells do this, would cancerous stem cells do something similar? Would they hijack this pathway? That was one. And then two, if quiescence is linked to immune evasion, could this be something that dormant or quiescent cancer cells would co-opt to escape from the immune system? Because we know that, for example, in prostate cancer or in breast cancer, some cells, some cancer cells can hide in tissues for not months, for years, for even decades, and stay in that quiescent state for this super long time, and the immune system doesn't see them, doesn't kill them, which means that somehow they have to manage to escape, right? They remain invisible. So can I use this previous knowledge and my tools to investigate these questions? And that's how I came here and I started my lab. And when I started my lab, one of the first students I had was Pilar Valdomino. She's also from Spain like me. She actually decided to join my lab after I gave a talk back in Spain. And she came here, she left everything and came here to work with Jedi T cells. And the first question she had was, well, can I use GFP and Jedi T cells as a model to study immune evasion in breast cancer? We were both of us very interested in breast cancer. So what she did is she developed this triple negative mouse model with GFP, which was basically 427, D2A1, EMT6, these conventional triple negative breast cancer cell lines. She engineered them with GFP. And now she could use Jedi T cells to investigate mechanisms of immune evasion. And her very first experiment, this is what happened. This is how it went. She injected, right? She had these mice with these green tumors growing, and then she injected Jedi T cells. And in the untreated mice, tumors grew very large. In the Jedi treated mice, right, you inject T cells that should kill all the tumor cells. The tumors shrunk a little bit, but not completely. And actually they went back to growing, not as much, but they grew a pretty large too. And she came to my office, oh my God, Judith, I think your T cells don't work anymore. I was like, okay, let's take a step back. Let's see what's happening. And when she sectioned these tumors, the ones that had been treated with Jedi T cells, she realized that actually a lot of the tumor cells that grew didn't have GFP anymore. And it was very obvious, right? Because we had a platform in which we could see the antigen. And what this was telling, I mean, this is not right, like, this is not something unknown. We didn't discover anything unknown. This is immune editing. Our advantage was that we were able to see it, right? So this tumor cell here grew because it didn't have GFP. And that's how it had this advantage. So now we thought that we had a very neat model to study specifically the ones that still have the antigen and don't get killed, which would be these ones, right? Like this tumor cell here has to be doing something to not get killed by T cells, as opposed to here. If I didn't have GFP as an antigen, if I was right, like blind to what antigens each tumor cell has and the antigen specificity of all the T cells in the tumor, and I took the tumor and I tried to study what's happening, I would be mixing mechanisms of immune evasion. But now we could distinguish. So how did we do it formally? So what she did is she took GFP tumor cells and she mixed them with cherry, which is not immunogenic in biopsy. And this was just a way to be able to separate targeted versus non-targeted tumor cells. She injected them into the mammary fat pad. And once there was a tumor, she treated these mice with anti-GFP Jedi T cells. And Jedi T cells did what they are supposed to do. They killed a lot of GFP expressing tumor cells. But no matter how we did it, we always had GFP cells that survived. And then our question was, how are these GFP cells that still have the antigen, how are they escaping from T cells? So we FACS sorted these GFP cells, we FACS sorted the cherry cells, and studied them. The first thing, obviously, that we looked at, coming from where we came, was, are they downregulating class 1? Because if you are a cell and you want to escape from CD8 T cell killing, the very best way is to downregulate MHC class 1. And it made sense. This could be a mechanism that, right, they would hijack, for example, from what we learned from the stem cells. And we know this happens in cancer. But actually, this was not the mechanism we found in this model. They don't downregulate. You can see here. They don't downregulate MHC, and they don't downregulate any other gene in the pathway. Because we checked in the RNA sequencing data we got back. So what would be the mechanism? Something before I go into the RNA-seq sequencing data, something else that we observed was that we had injected Jedi T cells that we were able to follow because they were CD45.1, so they were congenitally marked, was that they recognized GFP. So you would expect them super abundantly around GFP expressing cells and to a much lesser extent outside. And actually what we saw was the opposite. It looked as if these T cells were somehow trapped in regions where their cognate antigen was not present, and they were less abundant where GFP was expressed. So it could be some sort of T cell exclusion. So how could all of these be driven? You always get these RNA-seq data sets expecting that you're going to find the mechanism, right? And what you find is a lot of genes and a big headache. But some of the things we found to start with was that a few of the top upregulated genes in the GFP resistant cells compared to the, let's say, bulk of tumor cells that had not been targeted, which would be the cherry cells, is that some of these genes were markers of so-called cancerous stem cells. So genes that had been reported in breast cancer as the ones that label cells that had either higher tumor initiation potential or cells that had stronger abilities to resist chemotherapy. And we thought it was very interesting that by applying pressure with the immune system, these genes would get enriched, right? Because chemotherapy resistance versus T cell resistance, they don't necessarily have to go through the same pathways. We then took an unbiased approach. So what Pilar did is she got gene ontology terms, and by adjusted p-value, the ones by far that were the most significant, absolutely every single one of them had to do with cell cycle. So if anything, it looked at a very striking, obvious feature of these GFP resistant tumor cells was that they were slow cycling or in cell cycle arrest, which was very interesting because what I had found out as a postdoc was that stem cells are immune evasive only when they are quiescent. So it seems like cell cycle has to do with the ability of cells to escape from immune attack. So, but this is RNA, so it's really cell cycle different. These are just some of the genes. So what Pilar did is she had these, again, GFP and cherry tumors. And when she treated these mice with Jedi T cells, what she did is at the same time, she treated mice with EDU, which is a thymidine analog that gets incorporated when the cells proliferate. So when they go through an S phase, the cell gets labeled. And she treated these mice for 6 or 7 days. I'm not sure now, I think 6 days. For 6 days. And as you would expect, almost all the cherry cells, right? Because that's what tumors do, they grow. They got labeled. Conversely, almost none of the GFP cells that survived, right? Like the ones that had escaped from Jedi T cells had incorporated EDU. And this is the quantification. So this means that all these GFP cells that you see here, they did not proliferate not even once during the entire course of EDU treatment in a mouse model in which tumors grow for like 2 weeks. So half of the time, which is long for the tumor, when the tumor is growing exponentially, these GFP cells actually remained quiescent for these many days. So we thought, okay, quiescent cells are immune evasive. How can we study them? Because if we inject EDU and we use EDU to label quiescent cells, we have to kill them because you have to fix them. So how can we see quiescent cells alive so we can get them back and study them? That's one. And then two, can we study quiescent cells before we inject the Jedi T cells? Can we see what they do to stop these T cells before they come in? And after trying a lot of different reporters and, labeling with dyes and what, I mean, you can imagine, we found this construct that had been published, and it's a fluorescent reporter, a very strong fluorescent protein in yellow called Venus that is fused to a truncated P27 that is non-functional. So this P27 doesn't do anything, but gets regulated at the same time as the endogenous P27. And P27 is very high during G0 when cells are quiescent. So this reporter basically allows to see in yellow, in very bright yellow color, cells that are quiescent. This is what they reported in the paper. So we got this construct, we engineered our breast cancer cells, and the first thing was to try whether this construct really works in our model. And as you can see, in magenta, we have Ki67, and in yellow, the Venus P27 construct, and you can see very nicely how they anti-correlate. So indeed, this Venus reporter was able to label and would allow us to isolate and see live cells that are quiescent. The next question was, well, quiescence, what does it mean? Is this cell quiescent now, but was it quiescent 5 minutes ago, 10 minutes ago, 10 hours ago? Because we knew that our quiescent cells were quiescent for many days. So what Pilar did in this case is that she took tumors that expressed this reporter, the Venus P27 reporter. These tumors weren't treated, so they didn't receive any Jedi T cells. And she treated these mice for 1 week with EDU. So once the tumors were established, she treated with EDU every day during this exponential phase of tumor growth. And what she saw was that, you can see here, many tumor cells got labeled because tumor cells proliferate. But then these P27 cells actually did not get labeled, not even, like very rarely. So this means that, for example, this Venus cell here was quiescent for at least 1 week in a tumor that takes 2 weeks to grow. So that's a long time. And this is what we were looking for. Now, very long story short, what Pilar did is she engineered these cells switching colors around so she could directly test whether or not these quiescent cells get killed by Jedi T cells. So in this case, it's a tomato, which is red P27 reporter. And then she had cells that had this reporter and were GFP, and cells that were not GFP. And to start with, all of them had the same percentage of P27 quiescent cells. But when you inject Jedi T cells into these mice, you can see that there's a huge or a significant enrichment in P27 in these GFP surviving cells. So when you put pressure, the few T cells that survive are mostly quiescent, right? It's almost 80% of them. But then the question is, okay, quiescent cells in the tumor in this triple negative breast cancer model escape T cell killing. How do they do it? And then we went back to what we had originally seen, which is they seem to exclude T cells around them. At least that's what the very strong model of Jedi T cell killing was suggesting. We went back to the untreated mice, and it's no, right, like it's not nothing new if I say that T cell distribution within a tumor is heterogeneous. We all know this, right? That T cells are not evenly distributed in the tumors. And that's what many of us are interested in. How, right? Like what's keeping T cells away from some regions, and how do we get T cells to go everywhere inside the tumor? So this is how one of these tumors looks like. You have regions with more T cells, regions with fewer T cells. And then when you overlay the P27 reporter to see these quiescent cells, you see that actually very nicely, the region where there are few T cells perfectly overlaps with this fluorescent reporter for quiescent cells. So it looks like as T cells don't like to enter into this quiescent cancer cell niche. And I'm not going to show all the graphs, but we quantified in many ways. And it's very clear that T cells are much more abundant outside regions of quiescent cancer cells than inside regions with quiescent cancer cells. But this is a mouse model. Is this something that happens in people? Because otherwise we don't care, right? So Pilar had access to some biopsies from de-identified patients, pre-treatment, of course, and she could very nicely see, and these are just some examples, that so Ki67 cells are pretty abundant in triple negative almost everywhere, but in some regions, and in particular in some patients, you can see a significant amount of P27 positive cells. And when that happens, there are almost no T cells or very few T cells, as opposed to what happens when there are no P27. When a tumor has very few P27 or in regions with very few P27 quiescent cells, then you start to see more T cells. And this is what she quantified here. So indeed, it looks like this happens as well in patients. And when we are in the, as Stephanie said, we are in the cancer immunology department. So when Pilar presented this, some questions that people had, we are all immunologists in the audience, was like, but if they are quiescent, are they really dangerous? What does this mean, right? Like, is this, right? Because you theoretically want to kill the ones that grow. Those sound like the most dangerous ones. If cells stay quiet, do we bother killing? Is it a big deal if we don't kill them? And this was one question. Another question was, well, are they terminally arrested? Are they going to stay quiescent forever? And to answer these two questions, what Pilar did is she took advantage of this fluorescent reporter, because as a fluorescent reporter, you can FACS sort, you can isolate cells that are alive, and you know they are quiescent. So she took these tumors that express cherry everywhere. So cherry is the tumor marker. And then venous are the ones that are quiescent, and venous negative are the ones that are proliferative. And she re-injected FACS sorted cells, 500 of each one of them, into new mice. And to our surprise, not only did they grow, but they actually grew much more efficiently than the proliferative counterparts. So if you take a proliferative tumor cell from a triple negative breast cancer mouse model and you put it back, you need to use many more to get the tumor to grow than if you take a quiescent cell. So the quiescent cell was much more efficient to grow tumors. And we found this using different cell lines. So it was not just some flaw of one specific cell line. So definitely we care about these cells, because even if they are the only ones left at the end, when they are left at the end, they have the ability to regrow the tumor. So what can we learn about them? Because as I said, they have this fluorescent reporter, we can sort them, we can sequence them. That's what Pilar did, she sorted them. This is the bulk RNA sequencing analysis. This is a heat map of some of the genes that were differentially expressed. And you can see that, again, we found some genes involved in chemoresistance. And we found also genes, right, like SOX4, SOX9, that were involved in stiffness. So this fits with the phenotype of being able to regrow tumors. What else do these cells have, these quiescent cancer cells? Well, in this case, obviously, the first signature was cell cycle arrest, but the second signature that we found across the board was always hypoxia and glycolysis. So these cells seem to be highly hypoxic and glycolytic. And in the literature, there are reports saying that cancer stem cells are highly glycolytic. We go back, so this was great, but was not really helping us. I mean, yes, hypoxia could be immune suppressive, but how is all of this really working? And the problem is, right, like we are not comparing one tumor versus another. We are comparing sub-regions, right, like regions within tumors with each other, and that's tricky. So we went back to our histology and we had two questions. One is, why is this T cell not entering here, for example? Why aren't these T cells going in? And the second question was, okay, it's not that these regions are empty of T cells. They do have T cells. There's one here, one here. Why is this T cell not killing? These were the two questions. And nowadays, we all love to do single cell RNA sequencing. Every single paper has single cell sequencing, but single cell, it's great. It allows you to see heterogeneity, subpopulations. It's extremely informative, but it wasn't helpful here because we needed to do single cell sequencing from this niche and single cell sequencing from this niche. How do you do that? And I have to say that I was very lucky to have Pilar as a student because she's fearless. And we had this idea together, and actually together also with Olga Barreiro, who's an extremely talented and very smart postdoc in Uli von Andrian's lab at HMS. And the three of us had this idea, and Pilar really, fearlessly went for it, even though it was pretty crazy at the beginning. So we had this idea to be able to do single cell sequencing from specific niches within the tumor. And we call this technique photoconversion of areas to dissect microenvironments, or PATMESIC, just to follow with the Star Wars saga. So the way this works is Pilar had this mice called CAIDI. These mice express everywhere this protein, CAIDI, which is green. And it's a protein that can get photoconverted, which means that when you shoot UV light, the protein changes color. It goes from green to red. And this means that the cells to which you shoot the UV light, they go from green to yellow. And it's yellow and not red because the cells get a mix of green and red protein. So she had these CAIDI mice that are commonly used to track cells. And she grew tumors in these mice that had no color except for a tomato P27 reporter. So this means that the micro niches with quiescent cancer cells, these cancer cells would be red. She had these tumors, and she found a way to section approximately 100 to 200 micrometer thin sections from these tumors that were alive. And she obtained consecutive sections. And in these consecutive sections, what she did is then under the microscope, it's very easy to see these quiescent cancer regions in red, and then regions with proliferative tumor cells that don't have color. So then under the microscope, very precisely, and this is beautiful because it can be extremely precise. You can photo label if you want one cell. You can do any shape you want. It doesn't matter the shape of the niche. So what she did is she photoconverted every single cell that was infiltrating, right? Because in this case, infiltrating cells are green and come from the recipient mouse, right? So endothelial cells, fibroblasts, all the immune cells. So she photoconverted these cells inside the quiescent cancer cell niche. And in a consecutive slice from the same tumor, she photoconverted the infiltrating cells in the proliferative regions. Then she went to the sorter, fax sorted, and submitted for single cell sequencing. And now for the first time, she had a collection of, right, cells that came from one niche and cells that came from another niche from the same tumor. And she could compare how different these populations were, not only in terms of identity, but in their phenotypes. And this is quickly how this looks like under the microscope. So this is one of these niches. The tomato cells are all the P27 cells. So this is a clear niche of quiescent cells. And then she draws, right, like her region of interest, and every single cell inside the region that is infiltrating goes from green to yellow. And now at the sorter, it's very easy to sort these yellow cells. In another slice, it would be the opposite. So this is the data she got out of this first experiment that we actually repeated several times, and then she pulled different experiments because we didn't want to have bias by, right, like the microscope that day, the sorter that day, the mouse that day, the tumor that day. So this is pulling data from various experiments. So these are the different immune cells and infiltrating cells that she found in these tumors. And the population that showed the most striking difference between one region and the other were actually fibroblasts. Fibroblasts had a very clear, very obvious suppressive phenotype in the quiescent cancer niche. So for example, these LRRC15, these fibroblasts were described by Shannon Turley very recently. And these are all the genes that these fibroblasts that Shannon Turley identified express. And these fibroblasts, she found, were able to predict response to immunotherapy. They know these fibroblasts exist. They don't know, or they didn't report where within the tumor these fibroblasts are. And we found that actually these fibroblasts reside next to the cancer, to the quiescent cancer stem cells. That's where these suppressive fibroblasts are. And the more cancer quiescent stem cells you have, the more of these suppressive fibroblasts you have. What about T cells, right? Like we, obviously then the next question was, what's wrong with them? Besides that you have very few, is their phenotype differently? We reclustered T cells. We identified 4 populations or 4 subsets of CD8 T cells, excuse me. And we could classify them as terminally exhausted, intermediate exhausted, proliferative, which are the ones that express Ki-67, and they are kind of like more, I guess, fit, and progenitor exhausted. And when we went to quantify where they are located, we realized that progenitor and proliferative are much more abundant in the proliferative regions of the tumor. That proliferative T cells there are more abundant, and the super exhausted, terminally exhausted CD8 subset were much more abundant inside the quiescent cancer niche. So we know that these T cells exist. Many labs, John Wary, Arlene Sharp, and Nick Haining, many labs have described these terminally exhausted T cells. But as far as I know, they have not reported where within the tumor they locate. What our data is saying is that these terminally exhausted T cells reside next to the quiescent cancer stem cells. And we didn't have enough with the single cell data. We wanted to functionally see that this was true. And we took advantage of multiplex immunofluorescence. In this case, it was not a lot of colors. It was only 9 colors that we used here. We used a machine called Codex that was developed by Gary Nolan. And this machine allows to see multiple colors at once. And as you can see here, right, you can see CD3 and TIM3. So TIM3 in this case would be a marker of these terminally exhausted T cells. Sorry that I didn't mention that. So these terminally exhausted T cells are the ones with the highest expression of TIM3, which is an exhaustion marker. So they are PD1 positive, TIM3 positive. And what Pilar and she in the lab, an amazing technician that we have in the lab, both of them, they counted the number of these super exhausted T cells in different regions of the tumor. And as you can see, they are very rarely found in regions of proliferative tumor cells and much more enriched in the regions of quiescent cancer cells. How are these T cells getting so exhausted next to the quiescent cancer cells? What's next? Because as you can imagine, we have a plethora of cell types here to investigate. So what we did is, what do we know about the quiescent cancer cells? We know that they have a very strong signature of hypoxia. So we thought maybe we can use hypoxia as our sort of flag to see which other cell is not working properly in these regions. Pilar designed a hypoxia score, and then she found that actually every single subset of dendritic cells had this hypoxia score, and they were the ones with the top hypoxia score. What are dendritic cells? Well, you probably all know that dendritic cells are antigen-presenting cells. And actually, dendritic cells, I would say, are the most important antigen-presenting cell. They are very potent antigen-presenting cells, and they are the only ones that can activate CD8 T cells. They are the only ones that can induce a cytotoxic immune response through a process that we call cross-presentation. And I want to just take 1 minute here for, to advocate for dendritic cells, because in the field of cancer immunotherapy, mostly by people that approach this from the cancer perspective, everyone thinks about T cells, which makes sense because T cells are the ones that do the job. They kill the tumor cells, but they need to get instructed. They cannot do it by themselves. And dendritic cells are immensely important. And this is a very old paper now, but it's one that I think illustrates very well the importance of these cross-presenting dendritic cells. So in this experiment, and I'm gonna try to go very quickly here, this is a mouse model of a tumor that responds to immunotherapy. If you have a wild-type mouse and you treat the mice, the tumor regresses. If you take a rat knockout mouse that doesn't have T cells, the tumor keeps growing because you give PD-1 and there's nothing, right, for PD-1 to work. There are no T cells, the tumor grows. If you take a mouse that is only missing cross-presenting dendritic cells, which are this ridiculously small population, the phenotype is exactly the same as in the rat knockout. So not having cross-presenting dendritic cells leads to a complete failure to activate CD8 T cells, and then the tumors grow exactly the same. So it makes sense if dendritic cells are actually the problem. What did we find about dendritic cells? Well, dendritic cells across the board, these are all the different subsets. In the regions of quiescent cancer cells, the phenotype was highly, I would say, dysfunctional. They didn't express important cytokines such IL-12 that is known to be very important for activation of T cells. They had a downregulation of co-stimulatory molecules such as CD81, and they had a very significant downregulation of antigen-presenting genes, which are really the most important for dendritic cells because that's what they do. So a dendritic cell that downregulates MHC class 1 and MHC class 2 is a dendritic cell that cannot present antigen to T cells properly. And these are pictures that Pilar took to prove, right, that this is actually real in a, right, like protein tissue level. She visualized here dendritic cells with CD11C. And then as a readout for functionality, she used MHC class 2. And as you can see, CD11C cells in regions where there are no P27 cells, so in regions in which tumor cells proliferate, these CD11C positive cells, they have a lot of MHC class 2. However, inside the niche of these quiescent cancer cells, here that you can see in yellow, in these regions where there are a lot of quiescent cancer cells, now CD11C cells, such as this one, if you see, has very low MHC class 2. And she quantified these and quantified in different ways, and it's very, very reproducible. So dendritic cells inside the niche are highly dysfunctional. And we think that this is mostly what is driving this lack of T cells in the niche and this T cell dysfunction. So how is all this orchestrated? How are quiescent cells doing this? Because ultimately it comes from the quiescent cancer cell. The top feature these quiescent cancer cells had was a very strong HIF-1 alpha induced signature. So we thought, okay, can we narrow down this to HIF-1 alpha in the tumor cells? And what Pilar did here to demonstrate whether HIF-1 alpha in the tumor cells was playing a role beyond hypoxia in general, because the issue is hypoxia affects differently different immune cells. So, for example, T cells that have increased activation of HIF-1 alpha are actually hyperfunctional. So it cannot only be hypoxia, at least not hypoxia directly in T cells. So anyway, what Pilar did is she took these tumors that have a stable form of HIF-1 alpha, which is constitutively active, and she grew these tumors. And what she was able to see is that a tumor cell in a tumor that is not necessarily all hypoxic, but a tumor cell that has more HIF-1 alpha is more resistant to T cell killing, quantified here, when there's HIF-1 alpha activation. And these tumors, actually, what they do is they limit T cell infiltration, they increase T cell exhaustion, and they also reduce DC functionality. So just very quickly to summarize, because I guess I'm running out of time here. So what Pilar found was that there are quiescent cancer cells in the tumors that they form clusters, they live in micro niches. So there's like an environment effect here. And this leads to reduction of T cell infiltration. The few T cells that do manage to enter are highly exhausted. Dendritic cells in those regions are highly dysfunctional, and cancer-associated fibroblasts are highly suppressive. And all this together leads to enhanced survival of these quiescent cancer cells that actually happen to be dangerous because they have higher tumorigenic potential. So we think that these quiescent cancer cells represent reservoirs of resistance to immunotherapy, at least in breast cancer, which is where we found them. And finally, I just want to thank everybody in the lab. Obviously Pilar, who has been like the superstar here driving the project, but all my other students who are extremely talented, my postdocs, my technicians, our collaborators. So Martin Hemberg helped a lot with the single cell analysis. Our collaborators in the breast cancer program, Beth Mittendorf, Jen and Nelly. My mentors, Kai Wucherpfennig, sorry, Arlene Sharp, Marcia Hagis, Gordon Freeman, Harvey Cantor, and Uli von Andrian. And obviously, I have to thank Stephanie, who has been really a huge support for me when, since I started my lab and throughout the pandemic, which has been a really challenging time, and all the funding agencies. Thank you. Maybe I can stop sharing now.
Hey, thank you so much, Judith. That was great. Stephanie and I will alternate to ask the questions that are coming into the Q&A. So I will start with the first question coming from Yilin Shi. So the question is, during your study of cell killing by GFP positive cells, have you observed loss of GFP in the cells that are supposed to be GFP positive? And what is the kinetics of killing that happen in vivo? And do you think that there are differences between tissue to tissue? And do you see these differences?
So I guess the question is in the tumor?
Yes, I think so. If you see in the tumor, cells are supposed to be GFP positive. So have you ever seen the regulation?
So the killing happens pretty fast. T cells are very fast. It depends on the tissue though. I could see more differences in the transgenic mice that express GFP than actually into the tumor. The kinetics, so these experiments that I showed today, the killing was always 1 week. So from the day that Pilar injected the Jedi T cells till the day that she harvested, it was 1 week. Then she actually repeated and waited for 2 weeks. And it was pretty much the same, meaning you don't get more killing. So this means that all the killing, like all the cells that could potentially get killed, got killed within 1 week. And in 2 weeks, there was no more. She didn't do shorter, so I don't know if 3 days would be enough. We never tried. I don't think so. But a few days should be enough. And then the question about how is GFP lost? I mean, I think we don't know if it's a fraction. So the GFP negative cells that I showed in the very first image, some of them could be because when you engineer the cells, you never get 100% of them, right, with your construct. And then when there's very strong pressure, the very few that don't have it can regrow. Plus, it's possible there is some silencing. So that's why we decided to go for the cherry GFP. So at least we had like a reliable, reproducible way of comparing the two of them.
Great. You have a question from Daniel Temeko, who says a fascinating talk and asks, does HIF1 alpha also explain the hair follicle stem cell survival, or do you think it's likely to be a different mechanism?
Wow, the HIF1 alpha in the hair follicle. So they don't have any HIF1 alpha induced signature. So HIF1 alpha is not what is controlling antigen presentation in the hair follicle stem cells. We know that a major regulator is it's not a transcription factor, it's one of these regulators that bind to transcription factors. It's called NLRC5. That is extremely low in these cells, but how that is regulated and how that is regulated in response to cell cycle or at the same time as cell cycle, we still don't know.
Yeah, we have another question from Sanidas, who is saying, great talk, and then is asking, do you see a difference in epithelial versus mesenchymal characteristics in quiescent cells? For example, ECA adhering regulation in quiescent cell, or can exist on both sides of EN spectrum?
So that's a very good question. That was one of the first hypotheses we had, that these GFP cells that survived were likely more mesenchymal, but we don't see an obvious mesenchymal signature, at least not in these cell lines, these murine cell lines. I do think that this is probably the case in humans, but yeah, no, we don't see, at least we don't see downregulation of icatharine and upregulation of, vimentin.
Great. All right, Patrick Anderson is asking, what happens to non-classical MHC class 1 on these quiescent stem or tumor cells? Is there any change in sensitivity to NK cell killing?
That's a very good question. So what happens with NK cells? So in the quiescent cancer cells, class 1 doesn't go down. If anything, it goes a little bit high, mostly when you put T cells in, when you inject T cells, because of course T cells produce interferon gamma, so the few GFP cells that survive have a lot of class 1. In the hair follicle stem cells, class 1 is downregulated, not only the classical, also the non-classical. It's not zero, it's downregulated. And then one question actually a reviewer had was the same one: what happens with NK cells? So NK cells you don't find NK cells in the hair follicle or in the epidermis, so it could just be that they don't go there. But when I took the hair follicle stem cells and NK cells together and you put them together and you try to see, they don't get activated and they don't kill them. And I think, and I don't have a proof, but my theory is that class 1 is not zero, it's still there, it's just very low. So it's low to a level, not class 1 only, right? Like class 1 plus all the other genes in the pathway, which allow for loading. So these cells need antigen-specific MHC, right? So if everything is so low, you don't have enough of the MHC with the right antigen for your T cell, but you have enough MHC, so NK cells don't kill you. And I think that's, and actually, as I was saying before, the master gene here is NLRC5, and it's a gene that you do find mutated in tumors. And I think when a tumor cell gets to mutate NLRC5, gets this neat level of class 1 that allows for escape from both T cells and NK cells.
So Dr. Yonhao Zai says, great talk, and then is asking, have you found any mechanism of how are cancer-associated fibroblasts recruited to the quiescent tumor cells?
That's an awesome question. No, we don't know. We don't know if they are recruited. We don't know whether they change the phenotype when they go inside. Yeah, we don't know. That's a very good question. That's probably one of the follow-up that we have to do. How we actually did not check the fibroblasts in the HIF1 alpha stable tumors. That's something we should do, if it's actually HIF1 alpha mediated or is something else.
Great. Okay, we have time for one more question from Patrick Strasser. Great talk, really nice results. Maybe he missed it, but have you tried to knock out HIF1 alpha in the P27 positive cells in order to check if that's the main escape mechanism?
Wow, yes. So we've not knocked out HIF1 alpha only in the P27. Actually, I would need to think how you do that. I cannot think of a way of doing it specifically in the P27 because it's not a pre-existing population. It's a population that arises in the tumor. What Pilar did, and I didn't show, is she generated HIF1 alpha knockout, so tumor cells that miss HIF1 alpha. And what happens is actually the opposite of what happens with the HIF1 alpha stable, which is pretty neat, that these tumors get massively infiltrated, you get disease with high class 2, and there's a lot of killing. So therapeutically, it helps to block HIF1 alpha in the tumor cells.
Great. Okay, Judith, we have several more questions in the chat. These will all be emailed to Dr. Agudo so she can answer them. So I apologize, we have a hard stop now, but this was a really great talk, and we are so happy to have you here today.
Thank you.